Allora, benvenuti ai colleghi canadesi, signore e signori, in questa sede dell'Università di Palermo. La sala in cui ci troviamo è una chiesa ormai sconsacrata, la chiesa di Sant'Antonio Abate. È una chiesa che è stata restaurata dalla nostra università, completata nel 2010 ed adibita a centro congressi. Oggi ci tenevo a essere presente per questa iniziativa che l'ERSU e l'Università di Palermo che hanno intrapreso circa l'internazionalizzazione dei nostri studi e dei nostri studenti. Da anni l'Università di Palermo e l'ERSU, che è l'ente strumentale, noi siamo l'ente che garantiamo, possiamo dire, vitto, alloggio e negli scambi anche la ricettività alberghiera dei colleghi, docenti e studenti che vengono a Palermo. E assieme all'università abbiamo intrapreso questa iniziativa di internazionalizzazione verso i paesi dell'est europeo, ma adesso verso anche gli Stati Uniti d'America. Proprio recentemente abbiamo firmato una convenzione con l'Università di Houston, con la facoltà di medicina, anche con la NASA, e alcuni nostri studenti e dottorandi si trovano già in quelle sedi per poter svolgere le loro attività e allargare la loro conoscenza. Speriamo anche che da questo incontro lo studente possa trarre, oltre che conoscenze, anche la possibilità di lavoro, perché questa è sempre una terra di migranti. È una terra di migranti che speriamo, per noi non è positivo che i nostri migliori insegni. Una volta sono migrati la gente disperata, i lavoratori, gli operai, i minatori, adesso migrano anche gli intellettuali. Purtroppo perché per noi è una violenza, però la nostra terra è stata sempre terra di migranti, lo sarà sempre. E noi speriamo di fornire loro un'educazione però di più alto livello, di più alto respiro, ma sono sempre migranti. E questo però non ci rallegra molto. Però noi speriamo che da questi incontri internazionali quelli che conseguono quelle esperienze che possano poi tornare utili, tornare anche in Italia da noi. Noi speriamo che da noi, dopo questo, anzi spereremo che molti di voi potreste tornare da noi in Italia. Questo è un poco più difficile perché i migranti sono più degli italiani in questo momento, se li contiamo nel mondo forse superano. E voi che venite dal Canada o dagli Stati Uniti, il professor Assennato, che è il delegato per il rapporto internazionale dell'Università di Palermo, assieme al rettore sono venuti ieri sera o ieri mattina da Rosario e da Buenos Aires e hanno scoperto che a Rosario e a Buenos Aires la maggioranza sono italiani e hanno fatto delle convenzioni con una nuova università di medicina di Buenos Aires, che era l'ospedale italiano, che è diventato università pubblica. E quindi per noi diventa anche motivo di orgoglio poter intrattenere con loro queste realtà. Io mi auguro che la vostra permanenza oggi in questa città sia, al di là dell'incontro scientifico della mattinata, sia anche positivo, perché vi faremo anche vedere, per il tempo che abbiamo, che è molto molto limitato, praticamente alcune bellezze di questa nostra città, ma anche di questa nostra università, di questo restorato. Infatti prima del pranzo faremo vedere delle bellezze che si trovano proprio qui accanto, oltre che la bellezza praticamente.
Allora, professor Buono, grazie mille per questo ricevimento. Grazie. Do you mind if we sit down or do we have to stand up? Do I have your permission to stay sitting? I'm getting yes, stay sitting down. Okay.
Can you translate?
Professor Buono, grazie mille per averci ricevuto qui in questa bellissima chiesa. Per noi è qualcosa di speciale questo, perché come sai il nostro Canada è molto giovane. Allora vedere delle cose così è bellissimo, che l'avete trasformato in un luogo di insegnamento, vi do il mio. I'll just say a couple of words before I'll translate what Dr. Buono said, okay? Per noi essere qui già una settimana è veramente un sogno. Di tanti siamo stati ricevuti in una maniera calorosa da tutte le persone che ci siamo incontrate. Il vostro sole è veramente splendido. La geografia è per quello che anche noi ci chiamiamo l'organizzazione o l'associazione del Mediterraneo, perché la maggior parte di noi siamo del bacino del Mediterraneo. E abbiamo tante persone della Grecia, eccetera, e quello che è stato bellissimo di vedere, questo è il passato che voi ci avete, e questo, la Sicilia è stato sempre un centro dell'incontro delle varie culture, e questo qui si vede, e non soltanto, e si vede perché ci avete tenuto cura, perché l'accettate, l'abbracciate questo. And even now, today, this is what we are. We're all representing institution of higher education. We have Palermo, McGill, University of Montreal, and Chantal from the University of British Columbia. So, what you said before, that we hope that the scientific involvement here today will be of higher level, but also that we will develop good relationships at a personal level, like right here with your friend, Dr. Haber, he's in pediatrics, he teaches, and he does electives. He does electives with students from outside Canada. So therefore, at the personal level, we can do a lot. When I get back, definitely, we hope that our own universities will be able to develop these close relationships with yourselves here in Palermo. Now, what Dr. Bono said at the beginning, obviously he gave us the welcome in this 1600 structure, which used to be a church. Now they transformed it into a center of education. He also welcomed us to see Palermo, which we'll be seeing later on. The unfortunate thing is that we had very little. Just driving through, you saw the many things that we can possibly see here. So therefore, in the short time, I think it's just not even a little antipasto, maybe a little touch of antipasto. Okay, so there's only one fault in the Italians, only a little tiny one. That once we start talking, we can't stop. So I'll take my Canadian part and I'll stop right here and I will introduce you to our first speaker. Now, the first speakers among ourselves we know quite well. He's a dear friend, endocrinologist, professor at McGill. For some of you that don't know George, Dr. Tsoukas, Professor Tsoukas, obviously he's an endocrinologist. He is director of the Metabolic Clinic. He's an appassionato of history, as you very well heard that on Monday. He's also an appassionato of art. He is a fantastic painter. Il professor Tsoukas è un appassionato della storia, perché così ha cominciato lunedì, ma anche appassionato di dipingere delle ottimi quadri. But his main passion, and recently, and we can all say it, is giving conference to family doctors. He always brings the latest. So now today he's going to say a recent anti-diabetes and maybe stimulating those little buggers of beta cells. So here we are, Dr. Tsoukas.
What I will do this morning, I will tell you the research that I'm doing in my department. And I'll keep it short because we're late. But the research we're doing in our department is very exciting because it has to do with the cure of diabetes. We, among thousands of other people around the world, we finally decided we must cure diabetes and not just treat it. Before I start, I just want to tell you that I'm not representing any pharmaceutical companies, and our research is done from our own private research fund and McGill University Research Fund. So, we've known that diabetes exists for almost 3,000 years. The first person who described diabetes was Hippocrates, who in fact came to Sicily 4 times, because Sicily in those days was a huge center of learning. They had 56 universities in Sicily. Anyway, and he described diabetes, which means going through. He thought there was an imbalance of 4 tumors in the body, and in order to treat, one had to exercise, diet, etc., which is basically the treatment that we give to type 2 diabetes today. It has not changed. Galen, who also was a physician 1,000 years after Hippocrates, who also visited Sicily numerous times, and was the emperor's physician in Rome, decided it wasn't the humors, it was the kidney. There was a problem with the kidney, and the kidney was losing the sugar from the body. It was only about 160 years ago that Langerhans actually isolated the pancreatic cells, the beta cells, that we all know, and named them alpha, beta, gamma, delta cells. The alpha cell secretes glucagon, and the beta cell secretes insulin. And then our Canadians, Banting and Best, in 1922, gave insulin to the dog, and this new era of saving people with type 1 diabetes mellitus, who used to die until then, began, but we still really didn't know what the mechanisms are, and how could we possibly, we've cured so many illnesses in this world, and yet after 3,000 years of knowing this illness, we have to find out exactly what is it, how do we treat it, and how do we cure it. Now, the last 40 years has been the advent of the incretins that we now use to treat diabetes. The incretins are hormones that come from the intestines. And we also know that people who have intestinal bypass surgery, type 2 diabetics, are often cured of diabetes. So there may be growth factors involved that stimulate the pancreas, to modulate the pancreas, and hence to control it. But, as I say, no matter what we're doing, diabetes continues to grow on us. It's amazing that there's hundreds of millions of people around the world with diabetes. The incidence of type 2 diabetes goes up 5% every year. And we know that diabetes is the leading cause of blindness in adults. It is the leading cause of chronic renal failure, and most people who get dialysis are diabetics. It is the commonest cause of vascular disease, of strokes in the brain, and of course cardiovascular diseases, bypass procedures, etc. And is also the commonest cause of amputations. So obviously we have a major problem to address, and we have to address it rather rapidly because it's costing society literally a huge chunk of our budget, or our medical budget, goes to diabetes. And as far as type 1 diabetic management is concerned, we know it's very difficult for the young patients to accept, difficult for the doctors to do. There's intensive treatment with multiple daily injections of insulin. We now have the insulin pump, but that's still not easy to use. We have tried for years and years to transplant pancreas. It may work in certain cases, but it's too complex. We have to take pancreas from either cadavers by donors. It's not something that we use for the millions of people who have diabetes. And we also have the artificial pancreas. But again, this is not an easy thing. And the pathways have been more difficult than we expected them throughout the years. So we obviously have to deal with an approach to either remission or cure of the illness. And the past 20 years, people have concentrated on two areas. One is stem cell or adult cell therapy introduced into the pancreas. And the other is pharmacological therapy to regenerate beta cells. This is where I'm involved. But this is not the embryonic stem cell therapy. This isn't very easy. There's few embryonic stem cells. There's potential for immune rejection and side effects of immune suppression. And there's possibility of even inducing de novo cancers. And technically, it's very difficult, it's very expensive, and in the United States, it costs about $50,000 to do this, so we can't use this on an everyday approach. Now, using a viral vector, exocrine cells were transformed to resemble beta cells and introduced into the pancreas. But this, even though it represents an important advance in biology, the drawbacks include a reliance on viruses to deliver genes to target cells that are not very easy, it's inefficient. And the beta cells that result, they're not islet cells. And they're vulnerable, they're naked to the pancreas, and they're vulnerable to autoimmune attack, which happens, and we don't seem to get very much out of this. So we must find a way to stop the beta cell destruction before the disease manifests itself, which is prevention. And if we can't do that, we should also find a way of keeping the beta cells alive. So the molecular mechanisms by which beta cells are killed or stay alive are now being deciphered around the world. Why do we want to restore the islet mass and function? Well, obviously because when people become diabetic, type 1 lose almost all their beta cells, and type 2 lose about 85%. And if we're going to cure diabetes, either one or two, we have to restore the islets. We know that even a small amount of endogenous insulin production makes a huge difference in patients' quality of life and as far as complications are concerned. So restoring islet function represents the most important unmet clinical need for diabetes. This is the DCCT trial that was done a few years back, and we know that even years later, people 13 and 14 years later, once we have very small amounts of insulin still left, even though most of their beta cells were gone, still did better as far as outcomes, which is retinopathy and nephropathy, are concerned. So is there hope for regenerating beta cells? I think there is, and I'm going to show you that. There's new data that suggests that we may be able to preserve beta cell function, one, by reducing the destruction, and secondary, more important, by stimulating new growth. What causes beta cell death? A lot of work has gone into this, and we finally realized that there's loss of immune tolerance towards self-antigens from beta cell. This is the first thing that happens, leading to the process of cell death. Interleukins, particularly IL-1, and its role in the beta cell has recently become prominent. It's produced by most cells in response to endotoxins, exotoxins, or other cytokines. It functions as a pro-inflammatory signal molecule and mediates the acute phase response in infection, inflammation, tissue, and trauma. So one of the first things that is affected in diabetes is this system of interleukins. Pancreatic cells express the highest density of IL receptors among all body tissues. And IL-1 is also involved in the regulation of insulin secretion during sepsis and fever. So IL-1 has a homeostatic role in the regulation of insulin secretion when people become ill. So we now, until recently, we didn't know what happened, but for the past 2 years, we finally have realized how diabetes is caused. But particularly type 2 diabetes. So people who live in areas of the world that are not so fortunate as Sicily, to have all these beautiful natural products and do not follow the Mediterranean diet, follow a Western type lifestyle. They eat bacon and eggs in the morning, and steaks, and this and that. A lot of sweets, junk food may lead to obesity. Obesity increases insulin resistance and stimulates genes encoding molecules involved in impaired beta cell compensation. This leads to increased glucose, increased fatty acids, increased LDL, and increased circulating cytokines. So as we've been talking the last few days about cytokines involve macrophage recruitment and IL-1 production. On the other hand, glucose itself, hyperinsulinism, secretion of amylin, ends to beta cell IL progression. So this in itself, the IL-1 production, as I told you, is the first thing that attacks the beta cells. And it leads to progressive beta cell failure and apoptosis and progressive metabolic deterioration. I must tell you, these occur over a period of years. They don't happen in a day. I'm not going to go because we don't have much time, but just to tell you that this is called an inflammasome activation. The mechanisms have been really well described through caspase 1, you go to IL-1, and through the glucose affecting oxygen radicals, you go again to IL-1. So, chronic hyperglycemia causes inflammation of the immune system and beta cell death. This is more in picture form. So patients with healthy but genetic susceptible beta cells. And we know, for instance, that there's a family history of diabetes, even type 1 or 2. If you screen the relatives, about 25% of them have genes in the HLA category that are not proper, and may eventually, if there's a problem, affect the beta cell function. So, genetically susceptible beta cells affected by viral infections, adipokines, or other insults lead to a diseased beta cell, which, unless you interfere, such as using an interleukin-1 receptor agonist, leads to further oxidative stress, pro-inflammatory cytokines, interleukin-1, and cell death. If you block them, you may improve the beta cell. So, this is the way one should look at diabetes, especially in type 1. And type 1, again, is coming up 5 to 6% increase every year, so we have a major problem with it. So, the way for therapeutic agents is to target the new onset type 1 diabetics by immunomodulators that control autoimmunity that can work in the beginning before the collapse of the whole system. So the NIH in the United States has spent quite a lot of money trying to find these immunomodulators. And around the world, in Europe as well, huge studies on what could cause possible immune disorders. One that's been looked a lot is cow's milk, other things like gluten, and many other things that children take that might increase their autoimmunity. Once you establish the diabetes here, the way things are now, we can't really help them unless we give them insulin. So I'm not going to go into this because we're kind of late, but just to tell you that there's immune interventions, and which actually have been going on now for the past 15 years, but unfortunately have not given huge benefits as yet. For instance, beta HCG was used, and other agents used, but nicotinamide has been used, but these were only in trials. But again, the antagonism, there are also anti-CD monoclonal antibodies that appear to arrest. They stop the T cells from attacking the pancreas, and people have been using this. There's a couple of clinical studies that are positive as well. And as you can see here, agents in the type 2 diabetes, the ongoing trials now are CP3 specific antibodies, GAP-based vaccines, and children have been given oral and injectable insulin to develop an immune response to insulin to improve their self-antigen role. That's an ongoing study. DNA, plasmin, and proinsulin being used. And also, as I was telling you, gastrin and GLP-1 is being used as well as part of this process. We really don't know how these work, but it's being used. What I'm doing here is that we've got some INGAT peptide that I'll speak to you a little later. So, going through the viral stem cell approach or the immune approach hasn't yielded a lot. So what we're doing now is we're basically honing down on pharmacologically induced islet cell regeneration, because we know that the cells around the pancreas will switch roles and they'll go back and forth. So this is recent discoveries. This idea is much more attractive and near-term for therapeutic proposition. The islet neogenesis approach involves stimulating progenitor cells resident in the pancreas into intact, presumably pan-normally functioning whole cells. There's no viruses involved, no genetic material, and it's not expensive. So, the last few years we've raised this question: can we convert adult cells in the pancreas to islet cells? And a number of researchers have found transcription factors that reprogram different pancreatic exocrine cells in mice into cells that closely resemble beta cells. And these beta cells are indistinguishable from endogenous islet beta cells. And they express genes essential for beta cell function and can ameliorate hyperglycemia by remodeling local vasculature and secreting insulin. I showed the slide of this before. We also know that growth factors such as EGF and gastrin also may work through the same pathway. However, we have not done any human studies on this because EGF is a non-specific growth factor implicated in carcinogenesis, and the ethics involved in this will not allow us to use this in humans, but it's used in animals. What has been done, though, a lot of work has been done on the incretins, the DPP4 inhibitors, and you have 4 in Italy, and also the injectable liraglutide and exenatide. And in animal studies, these increase beta cell mass. You can take rats and treat them with streptozotocin, make them diabetic. The green are islet insulin-producing cells. This is normal. This is the ones that were killed. And after getting a DPP4, you still will get insulin-producing beta cells. So there's a number of studies going on right now in humans, both in DPP4s and with incretins, to see if we can get new beta cell stimulation. I'm actually doing one of them, and I must tell you, some of the patients you refer, we give them Vieta or Victoza. I believe there's regeneration of beta cells. So, I won't keep you very long, but I just want to tell you the research that I'm doing. And basically, using this INGAT peptide, which is a 15 amino acid sequence contained within the native 175 amino acid INGAT. And if we look at islets in vitro, we can take human islet derived structure into neoislets that produce insulin, somatostatin, and they look like islets even after day 4 when they're incubated with INGAT. And we know there's a transcription factor necessary for beta cell maturation, and with INGAT, this factor is expressed. And even when we do control dogs, we know that it works on dogs as well. Now, the NLD mice have a notorious diabetic state caused by immunity, and the softener, which blocks interleukin-1, was used. And also, if they used in India, together, this combination therapy, even though the mice were severely diabetic and all therapy was stopped, they were able to survive for a year and a half with normal insulin production. The clinical studies done with INGAM, there's been one with 70 patients with type 2 diabetics and 2 with type 1s, and all show promise because they lower the glycated hemoglobin. We have done one with the first one with type 1 diabetes. This one that shows a highly statistical significant effect of endogenous insulin secretion. And right now, when we go back, we're starting this very important study to test the use of anakinra, which is IL-1 blocker, in addition with NGAP in 16 type 1 diabetics, to see if this holds true as it did with the mice. It looks very promising and very exciting. So I'm going to stop here because we're running late.
So, to talk to us about the Mediterranean diet, we had Dr. Pasquale Mastronato, Director direttore della scuola di specializzazioni delle malattie cardiovascolari e delegato dei direttori per gli affari internazionali. So, a little afterwards, maybe we'll sit down and chit-chat together at those things. So, here's Professor Assem.
Professor Legata, Director of the University of Palermo, I welcome all of you, and I thank you for really choosing Palermo for an important meeting. Let me say that I'm very delighted to be here with you this morning, and as a Vice Rector of the International Relationship, to improve our training and our knowledge, and project program, some real, not only paper, but real cooperation between our institutions. Now, nutrition and cardiovascular disease, the benefit of the Mediterranean diet. I'm sorry for my English, but perfect. I hope that you can understand me. Well, the prevalence of the cardiovascular disease increased quickly during the first half of the 20th century as a result of the advent of the antibiotics that advantage the infection disease. During the 50s, important epidemiological observations like the fungus della and the Mr. Plate had highlighted the relationship between alimentation of the blood pressure level as an expression, possible expression of the alimentation, and incidence of the cardiovascular disease. In the same period, the Southern Country study, a major epidemiological study that addressed certain region of the world, correlated the incidence of the cardiovascular disease with the alimentation habits, noting that the Caribbean area has a low incidence of the cardiovascular disease in comparison of the low Europe and the United States. And all of us know that the difference in the alimentation, the amount of fatty acids in the diet, in the Mediterranean region, and the North Europe and the United States. From this observation, the Mediterranean diet, the alimentation, became the source of journalistic publication, but scientific observation, this observation were also sources of the scientific situation. For instance, 4 years ago, the British Medical Journal published its meta-analysis that showed the reduction rates of mortality from cardiovascular disease associated with combined decrease in adult school for Mediterranean diet. And Dr. Case, the curator of the semiconductor study, better demonstrated through his papers the relations between diet and the semiconductor. But what is the Mediterranean diet? The Mediterranean diet is a modern and recent recommendation inspired by the traditional diet. The Mediterranean diet is principally a plant-based diet. Every day, the Mediterranean diet consists of olive oil and meats, the abundant use of herbs and spices, water, and wine during meals.
Good one.
Also, of fish. The Mediterranean is a sea which is rich in fish, and to this, the Mediterranean well, we need to eat this food for the winter. The intake of diet food are forbidden. Well, it is the diet period, Mediterranean diet by Palermo, the second world. All of us know this parameter, I'm sure. Yogurt, cheese, and a low amount of pork consumption, high consumption of grains, high consumption of refined cereals, high consumption of wine consumption. Despite its name, the Mediterranean diet is not an official diet in the Mediterranean area, but is a nutrition of France. A large butter are commonly used in cooking, and the olive oil is reserved for dressing salad and cooked vegetables. In North Africa, wine is traditional.
They stimulate the transcription factor J and K, the nuclear factor kappa B, inducing an increase in the insulin resistance. Direct pull of lipid pull is depicted here in the same way if you want, because we start always with a state of hyperinsulinemia. We are, in other words, we are talking about the obese people or over-nutrition, people with overweight or obese or overweight. There are elevated regulatory factor and the nuclear regulatory element by the protein, which increase the expression of LDL receptor on the surface of the hepatocytes. This increase the uptake of also by the microRNA-33, that is also an impairment of the beta oxidation of the free fatty acid. So, as a net result, we have an increase of free fatty acid, also an increase in free cholesterol and cholesterol ester, which stimulate these lipid molecules are able to kill
which is the role of this protein HS60 in the pathogenesis of human disease. And this is by Dr. Francesco Palermo, professor of anatomy.
Professor Maiolo, Professor Bob for organizing the conference today. And I will start introducing you some new concepts about the chaperoning system and the chaperonomatis. Well, I think, I suppose that many of you already know that molecular chaperones, and I know which are chaperones, are a backbone class of molecules in our body. They are highly conservative in evolution, and the wall complement of these molecules has been recently called the chaperoning system. Their main functions are cellular functions. They are important to assess the coverage volume, graphic degradation of our cellular proteins, as well as protect other proteins from maturation after stress conditions. Since they are so important for cell tissue homeostasis, they have three groups. The first is by defect, for example, due to gene mutation. The second is by excess, due, for example, to gene dysregulation or post-translational modifications. And the third one, probably the most interesting, is by mistake, in which normal chaperones contribute to disease pathogenesis, like, for example, some form of tumors or autoimmune disease. Well, what we studied in our group in the last 10 years is this molecular chaperone named HSP60, is a mitochondrial chaperone that works together with its co-chaperone HSP10 in assisting the folding of other mitochondrial proteins. HSP60 is crucial for cell growth and tissue differentiation. Indeed, HSP60 activation results in early embryonic mentality, at least in mice. Now I will present you some forms of HSP60 related chaperonopathies that have been described in the literature, and we also contributed in their discovery. I start, of course, with HSP60 related, with two forms of HSP60 related chaperonopathies by defect. They both are neurodegenerative disease due to HSP60 gene mutations, and these diseases are named hereditary spastic paraplegia 13 and CHAP60 disease, in which we have the first form an axonal degeneration and the second one demineralization. So, HSP60, the conclusion is that HSP60 is a molecule with crucial roles also for central nervous system homeostasis. Then I present you some data from our group about a form of chaperonopathy by excess. This is a chronic obstructive pulmonary disease, COPD, that is known as a leading cause of mortality and morbidity worldwide, characterized by progressive airflow limitation with an abnormal inflammatory response and an increased number of inflammatory cells, in particular neutrophils. In the past, we already found that HSP60 and 10 levels decrease during the carcinogenic steps of airways, and also that HSP60 and 10 levels correlate in the inflamed mucosa of smokers with COPD. This prompted us to perform another study to determine levels of a panel of each of proteins, their main transcription factor, and a panel also of inflammatory markers in bronchial mucosa biopsies obtained from patients with COPD at various stages, comparing to smokers and non-smokers drugs. In birth, we found that both in epithelium and in lamina propria, HSP10, 40, and 60 levels increased in very severe patients compared to both smokers and non-smokers control. And in addition, that HSP60 levels correlated with elastase positive and mieloperoxidase positive neutrophils, both in osmolar subjects and in COPD subjects alone. And this prompted us to perform another study, an in vitro study, using already published model of COPD, so stimulating human bronchial epithelial cells with hydrogen peroxide at different concentration. And we found that these stimuli with hydrogen peroxide determined an increase in HSP60 levels in terms of protein and expression in terms of mRNA, and also the release, the secretion of HSP60 from these cells. Then we performed in silico analysis to research for putative binding sites for NFKB P65 that is already known that has a role in the pathogenesis of COPD inflammation. Putative binding sites for NFKB in the HSP60 gene. And we found 3 binding sites, one of which was the promoter. So we investigated further by the chip assay the binding site of P65 the promoter after hydrogen peroxide stimulation, and we found that indeed after the stimulation the binding of P65 to HSP60 promoter increased. So in conclusion, HSP60 levels increased in the mucosal subjects with the COPD. This increased FPU to oxidative stress that is present during the pathogenesis of this disease, and P65 is responsible in part for HSP60 gene upregulation, and finally HSP60 released from bronchial epithelial cells may play an active role in bronchial inflammation, for example, stimulating neutrophils. Then moving in the last part of my presentation, showing you some results about some form of chaperonopathies by mistake. The first is cancer. Actually, this slide is just to show you that HSP60 may have a different behavior in cancer, since it may be downregulated, as I showed you first in the airway, or overexpressed, like I'm going to show you in lung bowel carcinogenesis, in which we found a gradual increase of this protein through carcinogenic steps. And HSP60 may be also useful for diagnosis and staging of this form of cancer, since it may help detect vascular or neural invasion, lymph node metastasis, as well as neurogenic islands. And what is interesting is that in cancer cells in which HSP60 is overexpressed, HSP60 goes out from mitochondria, so it may be found in extra mitochondrial sites, like for example cytosol and the cell membrane. And it gains other extra chaperoning functions, like for example, cytosolic HSP60 may have an anti-apoptotic function, since may bind pro-caspase 3, so inhibiting its maturation and apoptosis activation. And membrane HSP60 is a prerequisite for its secretion via a pathway that involves lipid grafts, multivesicular bodies, and finally exosomes. And the Golgi apparatus also participates in this secretion. And finally, exosomes may have HSP60, exosomal HSP60 may have some role in cells or immune cells or endothelial cells. So in conclusion, HSP60 may participate in cancer progression in various ways, for example, inhibiting apoptosis of tumor cells or favoring tumor growth by exosome-mediated mechanisms. That is what we are studying in this moment in our laboratory. And finally, as I told you in the first part of my presentation, each of proteins are very observed during the evolution, so we can find analog proteins with high similarity also in bacteria, like for example chlamydia trachomatis in particular, that as we found quite recently shares sequence segments that could serve as a cross-reactive epitopes for anti-HSP60 Roel antibodies. This is a list of anatomical sites and cells in which HSP60 has been found in particular stress conditions at the plasma membrane levels. So, in these sites and in these conditions, HSP60 may be an autoantigen during persistent chlamydial infections and may involve in the pathogenesis of a number of autoimmune diseases. At the same time, both human and bacterial HSP60 share sequence segments with other human proteins, like for example the isoform alpha-1 of the human acetylcholine receptor. And okay, these are the 3D models in which we found that these potentially immunogenic antigen epitopes are present in the extracellular loop of this subunit of acetylcholinesterase receptor, and also in the so-called MIR, major immunoreactivity region of this protein. So it may have a role in the pathogenesis of myasthenia gravis, of course. This is why we postulated what I'm going to show you in this cartoon. After chlamydia trachomatis persistent infection, cells may produce large quantity of chlamydia HSP60 that may generate autoantibodies anti this protein that, as I showed you before, may generate a number of autoimmune diseases. At the same time, these autoantibodies may recognize other proteins like acetylcholinesterase receptor, also generating other forms of autoimmune disease. However, since, as I showed you before, also tumor cells may present HSP60 on their plasma membrane, these autoantibodies may recognize this HSP60, so determining cell lysis of tumor cells and then survival of an unaware subject. So, in conclusion, HSP60 may participate in autoimmune disease pathogenesis in various ways, for example, through molecular mimicry and with bacterial Roel, or through cross-reactive epitopes present in other molecules. However, antibodies anti-HSP60 might stop incident cancer. In summary, HSP60 is a molecule with crucial roles for cell, tissue, and then organism survival. HSP60 defect, for example, genetically, is associated with severe central nervous system diseases. HSP60 excess due to gene dysregulation may be associated with inflammatory chronic diseases. I show you some results about COPD, but in our group we obtained also analogous results concerning inflammatory bowel disease. And extracellular HSP60 may repair or perpetuate inflammation, acting like a cytokine. This is under investigation. And HSP60 may participate in the pathogenesis of cancer and autoimmune diseases by mistake. However, antibodies anti-HSP60 may stop by mistake an incipient cancer. And this is to acknowledge all the people, also the people that work in this study. Thank you for your